The initial SpaceBorn United mission aimed to test an IVF support system in microgravity. A mini laboratory containing yeast cells was sent into space for this purpose. The experiment focused on assessing the functionality of the life support system. The system was designed to maintain temperature, pressure, and culture conditions within specific parameters. Although the yeast cells did not survive re-entry, the mission provided valuable data. The life support system's ability to maintain set parameters was successfully validated. ARTIS, assisted reproductive technology in space, utilizes a rotating disc with microchannels. The rotating disc creates artificial gravity to simulate different gravitational environments. Researchers can test the effects of varying gravity levels on embryo development using ARTIS. ARTIS helps determine the minimum gravity required for healthy mammalian embryo development. SpaceBorn plans to conduct mouse embryo experiments using Skyroot rockets. Skyroot rockets enable late-access loading of early-stage embryos. Late-access loading is crucial for early-stage embryos due to their short viability window. Early embryos lose viability if loaded too far in advance of the space mission. This highlights the logistical challenges inherent in space-based biological experiments. The global IVF market is large, but success rates have plateaued. Space experiments offer unique data unattainable on Earth. Varied gravity and radiation levels in space affect embryo stress proteins. Analyzing these effects could improve IVF processes. Improved IVF processes on Earth could lead to higher success rates. Microgravity in space disrupts the hypothalamic pituitary gonadal (HPG) axis. This disruption affects the secretion of reproductive hormones. Cosmic radiation directly damages DNA, leading to genomic instability. Genomic instability poses significant threats to reproductive health. Both microgravity and cosmic radiation contribute to reproductive health risks in space. Experiments conducted in simulated microgravity environments have revealed developmental delays in mouse embryos. Specifically, the transition from the morula to the blastocyst stage is significantly prolonged under these conditions. Researchers observed a reduction in the number of trophectoderm cells in embryos exposed to simulated microgravity. Trophectoderm cells are crucial for embryo implantation and subsequent placental development. The observed reduction in trophectoderm cells suggests potential complications in embryo implantation. These findings also indicate possible issues with placental development in space environments due to microgravity. Simulated microgravity inhibits the decidualization of human endometrial stromal cells. This inhibition occurs by suppressing the ACT signaling pathway. Microgravity also suppresses FOXO3 expression in endometrial stromal cells. The uterine environment might not be prepared for implantation in microgravity, even if the embryo develops normally. Ethical concerns emerge regarding human embryo experiments conducted in space environments. These concerns include questions about the regulations that should be applied to such experiments. The duration of embryo cultivation in space also raises ethical considerations. Current legal and ethical frameworks are insufficient to address these novel scenarios. The inadequacy of existing frameworks necessitates the development of new guidelines for space-based embryo research.